Germline gene therapy, a form of gene therapy that is the ability to possibly eradicate hereditary diseases and disabilities. However, as promising as this seems, we must assess further, as possible risks and complications are just as likely.
Gene therapy is an experimental technique which is used to replace defective genes with a functioning pair. This is not so simple, as any foreign genes tend to be destroyed by the cell, which senses that it does not belong. To solve this problem, doctors often use a vector to deliver and spread the genetic material, which is accomplished by using a virus, with its harmful traits removed. As an example, an adenovirus carrier may be used as it targets the liver and hence is useful in the treatment of conditions such as hemophilia, as it delivers the new genes and enables the production of proteins which aid in the blood clotting. This is all still in the research and development stages, however, and as yet has not been approved for general use by the public.
Somatic gene therapy is the method most developed and ready for clinical use. There are two distinct methods which fall under this category: out of body, which involves the removal of blood or bone marrow from the patient, which is then examined to separate immature cells to select the desired trait before being inserted back into the body to spread and multiply. In the body involves the use of a virus with its harmful abilities removed, which effectively means it acts as a carrier for genetic information. The virus is introduced into the host body to infect other cells, transferring the genetic trait throughout the body.
Another way of making changes to an organism's DNA, specifically to cure inherited diseases, is by germline gene therapy. The most recent and controversial example of such is CRISPR-Cas9. Germline gene therapy works by either adding or removing parts of the human DNA, mainly by cutting out parts of the gene sequence. If successful, it will be the most efficient and accurate way to edit DNA to target genetic deficiencies.
Germline gene therapy, however, differs from traditional techniques as it exclusively targets the reproductive cells in the body. This means that any changes made to the DNA will be passed on to future generations. While this allows for the correction of hereditary diseases, it raises many moral considerations and as such is viewed under different clinical and ethical lenses. Because of this, the use of germline gene therapy is currently universally banned by all research and national bodies.
The prospect of removing hereditary diseases and disabilities seems like a promising one. However, to properly understand why gene therapy is so controversial, we must first look at the arguments against such a technology.
As mentioned before, one of the main dangers of germline gene therapy has to do with the fact that it is hereditary, and any alterations will continue to be passed down through subsequent generations.
Such a controversial technique, I think that it is only fair to weigh the benefits of such a technology with the cost of its misuse, as well as to address the possibility of unanticipated consequences.
As previously mentioned, the prospect to irreversible changes to the human DNA is a major limiting factor in the adoption of GGT as a viable therapeutic technique. This point is exemplified in light of the controversy surrounding Southern University of Technology Professor He Jiankhu's work last year. He had decided to edit the genes of two newborn twins in order to remove the possibility of them contracting the AIDS virus, a trait which they had inherited from their father. As well as going behind the back of the university with no prior authority to conduct the research, the case incited international condemnation for its potential to adversely impact the girl's development later down the line.
Another moral argument raised is that we have no precedent to influence the development of newborns, as it infringes on the autonomy of the individual, despite being motivated by benevolence. Additionally, the prospect of selecting for traits deemed desirable could lead to those in society with disability and unfavorable traits being looked down on and ostracized. Some even argue that to arbitrarily determine the traits of a newborn could set a dangerous precedent, whereby the notion of designer babies becomes a norm.
And since the procedure is considerably expensive, it would enable those who are wealthy access and deny it to those of lower class. In practice, this would enable wealthy patrons to select traits which surpass the superficial, things such as intelligence and the ability to select against disease and disability, and would serve to widen the disparity between rich and poor.
There are currently several laws limiting the experimentation and use of germline gene therapy for reproductive purposes. In Australia, the 2002 Research Involving Human Embryos Act forbids experimentation on human embryos, especially with the intention of reproduction.
The Prohibition of Human Cloning Research Act 2002 bans editing of human embryos or any other purposes.
With the future rapidly approaching, we must ask ourselves if the benefits gained by ethical use of this technology outweigh the potential repercussions of its misuse. For, if the technology is allowed to reign unchecked, the consequences could be drastic and irreversible. On the flip side, if we are too conservative in our regulation, we run the risk of stunting the development of the technology and preventing meaningful progress to be made. One thing is clear, however, that we must be firm in our stance and push for international agreement on the issue. Only then can we ensure the ethical use of human gene therapy and prevent the occurrence of actions which violate this basic principle.